Good morning, good afternoon, good evening, wherever you are joining us from today here for our Food Packaging Forum webinar on can coatings. I'm very excited actually because this is our first webinar that we're holding, and so very welcome to all attendees. We are recording this webinar and we will make the recording available on our website after the event, as well as the slides that we're showing during the webinar today for your future reference. I'm very pleased to let you know that we have two presenters here with us on the call today. First of all, to kick us off, Dr. Birgit Goyke from Food Packaging Forum will talk to us about the new FPF dossier that she prepared on can coatings. And after Birgit, we have Dr. Maricel Maffini joining us from Maryland in the US. And Dr. Maffini is a co-author of a very recently published study on a novel can coating using the monomer TMBPF, and Dr. Maffini and her colleagues tested this monomer for estrogenicity, and she will share her findings on that. And after the two talks, we will have time for some questions and discussion, and I invite all participants of the webinar to join us for that. Please email us your questions during the webinar. You can just drop us an email on events@fp-forum.org, and we'll try to get to as many questions as possible during the webinar. Great. So just before I ask Birgit to take over, I just want to give you a little bit of background about the Food Packaging Forum. Who are we? Some of you probably know us really well, and vice versa, but others may not. So we're a charitable foundation established in 2012 in Zurich in Switzerland. And our task is to do science communication on chemicals in all types of food contact materials. So we look at how these chemicals migrate into foods and their impact on health. And we have a focus on current science, new scientific understanding, and I hope that you will find a lot of information that is useful to you on our website. And with that, I would like to hand over to my colleague, Birgit, who will share her slides right now. Okay, I hope they're coming up. And to you, Birgit.
Die Slides, vielleicht kannst du. And maybe you can make me a presenter, because I cannot do it on my own. Jane? So can you hear me? Okay, I got the feedback that we solved all the technical problems, and
Right-click.
okay. So, a second start this morning. I also welcome you very warmly to our first webinar. You see it's not that easy to start it, but now it should work. So I will present our latest dossier on can coatings, which was published just some few weeks ago. And Jane already gave a nice introduction into our work, and I just show you a quick screenshot of our also fairly new website. Here you can already see that the can coatings are promoted in the middle of the website, but we have other things which may be of interest to you. And for those of you who don't know the website very well, you can get a lot of daily information and latest news on the front page. So if you go there, there should be news available on every day or each day. And then we have also background articles. And there are two different kinds of background articles, and that's maybe not so familiar to all of you. So we have the section on food packaging and health, as you can see here. And these provide quite short summaries on topics which are relevant for the safety of food packaging materials. And then a little bit more hidden and available for a little bit more than a year now are the dossier articles. So these are much more detailed background articles, and you may take some more time to read them, but they provide you with a lot of useful information on certain topics. I will come to that later. And another, a little bit more hidden icon are the events. We have a yearly workshop at the FPF. You may have already joined it. Maybe you want to join in October of this year, so you find more information at the right corner of our website. Now coming back to the dossiers. We have published 11 dossiers since 2013, and the topics are quite diverse. So we have dossiers on certain chemicals, on bisphenol A and bisphenol S, for example, which are used to produce food packaging materials. Then we have dossiers on materials or groups of materials, or also groups of chemicals. Number 10, you can see the fluorinated substances, which are also commonly used in food contact materials. And then the latest dossier, which I will present here, is the dossier on can coatings. The dossiers, all of them have a quite dense content, so it's a lot of information hidden in the dossiers. And you may find some interesting information in the introduction about definitions, nomenclature, chemistry, which are basic to understand a certain topic. Then you find more applied information, as I would like to call it, on the use and production of materials or chemicals, market data, and then a lot of things which are related to the safety of food contact materials. So information on regulations, on migration, toxicity, exposure, biomonitoring, and so on. And last but not least, all the dossiers are providing a lot of references. If you're interested in even more detailed information, you will definitely find them there. So why have we decided to publish or write a dossier on can coatings? Let me start with the can itself. Cans are a very important food packaging material because they preserve food for many, many years. And regarding food waste, it's an important packaging type. You cannot use cans as such. You cannot use just metal cans. You need to coat them because otherwise the cans are not as resistant as they should be. And coatings protect them from the effects of the food, and they also prevent reactions between the can's metal and the food. So we, of course, also do not want to have any metal in the food which may disturb or destroy the food's properties. And a third important point is that the currently used coatings, which are almost exclusively based on bisphenol A, they are getting under pressure due to different reasons. So there is strong toxicological evidence that BPA is released from the coatings into the food, and so that it should not be used in coatings maybe anymore. Public discussions have started, and also regulatory decisions have been made which lead to the replacement of bisphenol A-based coatings. Coming back to the cans, I would like to show you some numbers. So there are 300 billion beverage cans per year, which are used globally. That corresponds to 40 cans per person and year. And most of them, the beverage cans, are made of aluminum, and only a small portion is made of steel. This doesn't matter so much because both of them have to be coated. So aluminum and steel cans have to be coated. Then additionally, 75 billion food cans per year are used, which corresponds to 10 cans per year and per person and year, and this is mainly, or to a higher percentage, is made of steel. So the global market estimates are 64 billion US dollars, and approximately 5% are represented by the coatings. Now coming back to the can coatings themselves. So why are they so important? They have to fulfill a series of requirements. So they have to withstand the can manufacturing process at first. Then they have to be universally applicable. So all different types of food are filled into coated cans, and the coating should not interact with these types of food. They should withstand the sterilization process. They should minimize the release of their constituents. They should adhere to the metal, because even if the can is formed, it can still happen that it's bended and still possible to use the can, and the coating should not sit on the food in the end. They should prevent corrosion, and last but not least, they should not change the organoleptic properties of the food. Here you see this blue arrow. This is the problematic issue I've mentioned before. The most commonly used coatings, which are based on BPA, the epoxy coatings, they do release problematic or harmful constituents into the food. So that's the reason why the discussion has started to replace them. Now coming to the epoxy coatings, they are in use since the 1950s, so more than half a century of use has been reported, and as I said before, they are the most commonly used type of can coating. They're generally based on bisphenol A. You can see it in the brackets. There you find the structure of bisphenol A. And they have high stability and protective function, good processability, low cost, so their overall features or properties are very good. There are many blends available, but they release certain chemicals into the food. Here you see the epoxy resin again, and what has been commonly found is the basic monomer BPA. And it has also been correlated that people who consume a lot of canned food have elevated levels in their urine or blood. Then other constituents are released into the food from the coating. One which is also quite well known is batch, which can then be transformed into hydrolyzed and chlorinated derivatives, which has been regularly measured in canned food. And then also cyclic derivatives of batch have been found, and they are consumed by the people who eat canned food or drink canned beverages. So coming back to BPA, because this is the best known migrant from can coatings, I will just show you why it is such a hot topic at the moment. So there are the replacement of these epoxy-based can coatings. There are risk assessments ongoing by EFSA. One has been published in 2015. You may not see the details here, but you can find the details in the dossier. Then there is another one ongoing by EFSA. Then there is an ongoing risk assessment by the FDA, named the Clarity BPA study. Then also regulatory measures have been adopted. So in California, BPA became a chemical which is listed under Proposition 65, so it has to be labeled when it is used. So cans need to be labeled in future that they contain BPA-based materials. Just recently, BPA entered the list of substances of very high concern, which is a list of hazardous chemicals defined by the European Chemicals Regulation REACH. And in France, BPA has been completely prohibited in food contact materials. This is based on a lot of toxicological evidence. Here are just 3 examples, but you find hundreds of scientific studies which describe the toxicity of BPA. And it also entered the headlines. So the public is informed about the problem of migration and that the safety of cans may be compromised by migration of BPA and related compounds. So that initiated the search for alternative coating materials. And here you see a list of material types. There are, I would say, hundreds of them under investigation. And in the dossier you find a list of advantages and disadvantages for each of these groups. It's definitely not complete because they may be also mixed and blended, and not all the information is available. But it seems as if none of these materials fulfills all the technical requirements as well as epoxy-based coatings at the moment. So it will be interesting to see what may be a suitable alternative in future. And another thing which has to be considered, also the alternative coating materials will cause migration. And migration does not only consider the starting or monomers, the starting substances or monomers, as it seemed to be for epoxy coatings in my slide, but also many other substances may migrate. So there may be oligomers, catalysts, additives, scavengers, cross-linkers, lubricants, and so on, which may migrate. So if new materials are used or will be used in future, this has to be investigated as well. So let me finish with some conclusions and also with open questions. So there is a high pressure to develop alternative coating materials. The technical requirements are demanding because cans need to be stored for a long time, and they are processed, and they may be deformed during production or during storage or transport and so on. But the market opportunities are favorable because the market is huge, and I think everybody would love to take a safe, cheap, good new coating. But questions remain, and many more questions will remain or will come up probably later on. But first ones are, how shall novel coatings be tested, and are they really safer before I introduce them to such a big market? Maybe we hear an answer in the next presentation by Dr. Maffini, and with that I thank you. If you have questions, we will find time to discuss them later on. You can send them to the following email address. And thank you very much.
Okay, great. Thank you, Birgit, for that. And I apologize for the technical difficulties that we had at the beginning of your presentation. Again, please, if you have questions, you can send them to events@fpforum.org. And I would like to invite Dr. Maffini to join us now. And I will stop sharing my screen so that you have the screen to yourself. Maricel, can you hear me?
I can. I cannot
Great.
see the presentation yet.
You can't see it? I think you have to pull it up yourself. That's what I've figured out now. So you can start sharing. I'm sorry about that. We rehearsed this, and of course, as always, it worked during the rehearsal, and now we have to improvise. But I think Maricel's gonna get us going in a moment.
Okay, it's uploading.
Great, fabulous. Good. And while we wait for Dr. Maffini's slides to come up, I would just like to introduce her briefly. Maricel has an academic background. She is, oh, here are your slides. Okay, great. So
Yes.
maybe you want to introduce yourself then. Thanks, Maricel. Over to you.
Sure. Thank you, Jane, and thank you to the Food Packaging Forum for this webinar and the opportunity to present some of this data. My name is Maricel Maffini, and I am a scientist and now working as an independent consultant. So today I want to share with you some of the information that was just recently published. And before I do that, I want to disclose that I was a paid consultant to advise the Valspar Corporation, who is the sponsor of this particular research, to advise them on science and testing strategies for a new polymeric can coating material that they had developed. So briefly, I will discuss the strategy to identify and to test the monomer that was used to produce this polymer with a broad spectrum of applications. And for more information, more details, I encourage you to read the article that was published a few weeks ago in Environmental Science and Technology. So as Dr. Goiker said, there's a challenge here because BPA has been a very successful monomer in producing epoxy resins that have a broad application and work on a variety of foods. And there are the challenges with the replacements is that they may work in some foods and some applications, but not on all of them. So it was quickly concluded that to replace a BPA-based epoxy, what is needed is an epoxy-type material. So what are the lessons learned from the BPA epoxy resin? So we know that BPA is an estrogenic chemical. We also know that BPA is used twice in the process to develop the polymer. We also know that there's this structure called, another disclosure, I'm not a chemist, so I apologize if I make some mistakes when it comes to the chemistry. I am a biologist by training. So we do know that the structure for 4'-methylene diphenol is particularly important to provide the performance of the epoxy resins. So again, continue to learning from BPA, we know that we do need this structure for the best possible performance. It provides a functionality that is unique, that is not available from other materials, and the polymers are flexible, provide high integrity, and work across the entire spectrum of metal food contact applications. Now, we also know that bisphenols are the only commercially available sources of the 4,4'-methylene diphenol. So you would say, so what? What's the problem with that? The problem is that bisphenols have, of those that we have data, many have shown to have estrogenic activity, such as like BPA. But the second problem is that the great majority of bisphenols available have little or no health effect data. So the challenge was to find a novel bisphenol that will lack estrogenic activity and at the same time impart the technical performance and integrity that is needed to ensure food safety. So, and additionally, if it's possible at all, that there is no residual bisphenol in the final product. As Bigot was mentioning, BPA is present in canned foods. So, continue learning from BPA. What we know through modeling and through in vitro testing as well is that the estrogenic activity seems to be associated with the flexibility of this methylene bridge. The more flexible the bridge is, like in the case of the para-BPA, the higher the likelihood that the molecule could bind to the estrogen receptor. If this bridge becomes less flexible, more immobilized, like in the ortho-para-BPA on the right-hand side here on the bottom, there is, in this case, a poor estrogen receptor agonist. The likelihood that estrogen that would have estrogenic activity is very low. So after screening of multiple bisphenols, it was found that tetramethyl bisphenol F was a potentially good candidate because it contains the structure needed to provide performance, but also through molecular modeling, the flexibility of the bridge was very poor. So that was encouraging news. So this is schematic representation of how the tetramethyl bisphenol F based polymer is synthesized. As you can see, although the name is bisphenol F, there is no bisphenol F involved in the synthesis of the polymer, and it's not a monomer itself. TMBPF is made of silanol, and then through this process, the DGA format is made that is needed then for the polymerization and the preparation of the final product. This is just a picture of how BPA looks like compared to TMBPF. So, so far we had the a polymer. It was we have the functional structure needed for its performance. So now we need to test whether the monomer has estrogenic activity. And the hypothesis was that based on modeling, it would not have estrogenic activity, and it would not migrate because, as I showed you earlier, is formed. TMBPF is formed and reacted immediately to form the polymer. So the expectation is that there won't be migration or residue. So if these two hypotheses are true, we still have to deal with the final product, with the polymer. That is what is actually in contact with the food. So we also decided to do in vitro testing of the final product and also an analysis of the migrants. So very briefly here, this was the approach to test for estrogenic activity. That's what EA means. And the migration assessment for the monomer and the polymer. There were in vitro and in vivo testing. Some of the testing was done in collaboration with academic laboratories, and there was also a quantification of migration and a profile of the non-intentionally added substances. So very quickly here, I will show you the data from the analysis of the monomer, the TMBPF. The first test was done using the e-screen. That is an assay that uses cells called MCF7. These cells are breast cancer cells that need estrogens to proliferate. And these are just pictures of how they look like, or what the e-screen assay looks like. So these are typical dose-response curves. We use estradiol as a positive control, so this is your typical estradiol dose-response curve. We use bisphenol A also as an estrogen, as a chemical with estrogenic activity. These dotted dots, the dark dots, is the BPA dose-response curve. And here at the bottom, this flat line is tetramethyl bisphenol F. So in the presence of TMBPF, the MCF7 cells that need estrogen to proliferate did not proliferate, which we concluded that TMBPF does not have estrogenic activity, at least in this particular assay. The next assay that was performed was an in vitro assay, also very common for testing for estrogenic activity. It's called uterotrophic assay. It uses pre-pubertal rats, female rats, and is based on the concept that these animals do not have estrogen in large amounts in their bodies, and in the presence of estradiol or a chemical with estrogen activity, the weight of the uterus increases. So this chart shows the positive control. The animals that were given ethinyl estradiol, the weight of the uterus increased manyfold. Neither of the TMBPF doses increased the weight of the uterus. There was an additional test was done in these animals, or analysis was done in these animals that it is novel. We also removed the mammary glands because mammary glands are sensitive organs to estrogen activity. We looked at the mammary glands and we didn't see any changes in the histopathology of the treated animals. The next test was the pubertal assay. In this case, male and female rats were used, and this what was done. We tested for the measurements were on endpoints of puberty. In males is preputial separation, and in females is vaginal opening. These animals are treated for much longer times. It's a month and a half for males and females, for females and males. And what we see is that there was no changes in the age at which the animals reached puberty. The animals treated with TMBPF and the control animals not treated with TMBPF all reached puberty more or less at the same time. There were no statistically significant differences. We also didn't see differences in the estrocycles of the females. There were no changes in the weight or the pathology of the reproductive organs, both in males and females. And also we looked at body weight, and there was no increase in body weight in these animals. We also looked at the mammary glands. Puberty is a particularly important time for the mammary gland because it's growing rapidly and developing, and it has many different structures that are features of puberty. We looked at the treated animals, you can see in the middle and the right panel, and compared them with untreated animals. We didn't see any changes in the developmental pattern of the mammary gland. So this led us to conclude that in both animal studies, TMBPF did not show estrogenic activity. Now, for the polymer, we did test the polymer. It was extracted with two food stimulants recommended by the Food and Drug Administration. 3% acetic acid, representing acidic foods like tomatoes and fruits, and 50% ethanol, representing water-based, fatty and alcoholic foods. So these extracts were then tested in the E-screen assay that, as I said before, is based on whether the cells will proliferate in the presence of an estrogenic, a chemical with estrogenic activity. So again, here these are the dose response curves for the 3% acetic acid, and on the right-hand side is for 50% ethanol. As you can see on the bottom, these are the dose response curves. They were flat, and there was no increased cell proliferation. To make sure that this was actually due to lack of cell proliferation and not cell death, additional plates were prepared, and the same concentration of estradiol was added to the plates to induce full proliferation, maximal proliferation. So these are the plates that have not only the extract from the polymer, but a single dose of estradiol, and as you can see, at least up to 2 parts per million there was no decrease in the cell proliferation, and then there was cytotoxicity observed at higher concentrations. So basically, once again here, we see that the extract from the polymer did not increase cell proliferation. There was a quantification done by FERA, that is a laboratory based in the UK, of the migration of TMBPF in the extracts, and also a chromatographic analysis of the NIAS from the extracts. This is a busy table, but what I wanted to focus on is on the right-hand side here, the TMBPF concentration in the extracts was below the limit of the detection of 0.2 parts per billion in all cases, both for the acetic acid, for the ethanol, and also for the extracts that were concentrated and then redissolving DMSO, that this step was needed for the extracts to be able to be tested in on the cells in vitro. So it seems that TMBPF is not migrating out of the polymer, at least at the level of detection that could be measured. Very quickly here, these are images of the non-intentionally added substances in the food simulants. The chromatograms on the top are for the blanks for both simulants, ethanol and acetic acid, and the bottom are the chromatograms for the extracts. In both cases, there were 650 masses detected in the ethanol extract and 651 in the 3% acetic acid extract. There was not an attempt to identify those masses. My last slide is just a summary. Our hypothesis that it was initially drawn from the computational modeling that TMBPF would not be an estrogenic compound was confirmed. All the data in vitro and in vivo, and additional data that you can find in the publication confirms the lack of estrogenic activity by both the monomer and the polymer. The migration of the polymer is below the limit of detection, and also this led us to conclude that human exposure to the monomer appears to be negligible. So with this, I thank you. If you have questions, you can send me an email. If you would like copies of the article, you can send me an email, and I send it back to Jane. Thank you.
Thank you so much, Maricel. That was excellent. Thanks a lot. Now, we come to the part that gets interactive, and so I would like anyone who has a question to make use of our email address, events@fp-forum.org. We will take the questions as they come in, but I also have prepared a few questions for our speakers, which I can also get to. And another thing that I'm going to do now is I've muted the audience, but I'm going to unmute you now. And if you would like to ask a question, then you need to unmute yourselves again, and then you can just speak up, and I hope we can hear you. Okay, good. I have a question for Birgit. Birgit, could you tell us how coatings are regulated? There's a patchwork of regulations in Europe. Maybe you can also have a look at what it's like in the US. Can you enlighten us on that, please?
Yes, Jane. As you mentioned, it's really a patchwork. So in Europe, there is no harmonized regulation. So it means the EU doesn't provide harmonized regulation for all member states, but there are national regulations. So there is, for example, one in Belgium, and the Dutch regulation on can coatings is currently under review, so I think there will be a new amended regulation coming up in near future. And of course, can coatings have to fulfill the requirements of Article 3 of the framework regulation in Europe, so they have to be safe. They should not endanger human health. They should not change the organoleptic properties of food. So that's the basic regulation in Europe. And then there are also some specific regulations on certain chemicals which may migrate. So there is a limit for BPA, which is quite new. Then there are limits for batch and its derivatives. There are limits for, let me think, for tin. I think for these materials or for these substances you have specific European regulations. In the US it's regulated under the Code of Federal Regulation, which covers polymeric. So the number is, I have to check that, I don't know it by heart, it's 175-300. You find all this information also in the dossier, of course, and that regulates polymeric and resinous coatings. And as I mentioned before, also in the US, you find specific laws or regulations. For example, in California, BPA has to be mentioned on a product if it's used. So cans need to be in near future be labeled that they may contain BPA, which may end up in the food. There may be some more, but these are the most important ones, I would say, at the moment.
Okay, great. So that means for TMBPF to become authorized. Okay, those are challenges of technology. I hope now you can hear me. For TMBPF to become used as a can coating, then, for example, it would have to go through a member state authorization in Europe, and in the FDA it would have to go through the food contact notification, right? Is that?
I assume so at the moment, yeah.
Good, thanks. Okay, so we have a question. We actually have quite a few questions. I am just trying to paste them here. So, Amaury Patin, I hope I'm saying your name right, has asked us about, this is for Maricel, I guess. So Maricel, have you also done some experiments on badge of TMBPF, so the epoxide form of TMBPF, and could you find these compounds in the migration studies? Maybe they were the NIAS, which you mentioned, Maricel, which were not identified. Can you tell us something about that?
Sure. I am not aware that badge was tested. I was not involved in that. And it's possible that some of the NIAs, the 650 that were identified, are badge, but there was not attempt to identify any of those. It is possible now with this research that has been done that others could continue researching on this particular topic or any other issue that people would like to look at. I want to mention that the full reports for the migration study, the 2 animal studies, another study done in vitro on transactivation of estrogen receptor, are available for anybody to look at. The full reports with raw data on the Valspar website that Jane, maybe you can then circulate.
Yes, we actually have the link up. As far as I'm aware, you can't download the report, so if you want to read it, you have to sit at your screen and read it there, but the information is there, and yeah, we have the link, and we can easily circulate that. Great, thanks, Maricel, and thanks to Amaury for the question. We have another question from Dan Steen from OI. To both speakers. So for our speakers, what is the next to be done in understanding the science or health impacts of can coatings? Is there additional science and development? What should we be looking for? Maricel, do you want to field that question first, maybe?
Sure. I think there's always more that can be done. The publication of this paper opens the door now for other people to do more research on potential effects of the monomer and actually to explore more what the effect of the polymer is, and to perhaps do more investigation on identifying all this, the denials. I think the concept, or what we were trying to do with this paper is also to show that nobody, not a single person or company or academic has an answer to a complex question like what are the health effects of can coating. That I think we need to start looking at collaborations and people with different expertise to really look at the potential health effects of food contact materials in general, and also make the data available so it can be shared and discussed across all the stakeholders. I think that is one of the important lessons here. But as I said, TMBPF is available for people to study it, and I think it would be great if this continues. This is just one step in the understanding of how this coating and this monomer work.
So, Marisa, maybe I can just kind of ask the question again, what should we be looking for? I mean, you've looked at estrogenicity now. Obviously, there's a whole bunch of other effects. There's a whole bunch of other hormone receptors. What would you recommend to look at now? Anti-androgenicity next, maybe, since BPA is known for that?
I know that there is data available from the company. Valspar had looked at different hormone receptors using mostly in vitro assays for receptors like dioxin and androgen. Those were not positive assays, so there was no activity from those. I don't know if they would publish that information. But I think, I think what I would like to see is people to look at more modeling of this particular molecule in systems. And based on that computational modeling, see which are the potential targets for this molecule in the body.
Okay, that's an interesting proposal. We've run over time a little bit, but I still, because we had technical problems, I still would like to ask Birgit one last question. So we've known for quite a long time about the problems with BPA, its endocrine disrupting properties. Why has it taken so long for new coating to enter the market, and will we see the same kind of time lag, 20 years maybe, until a safer coating based on TMBPF hypothetically would enter the market? Birgit, can you share some thoughts with us on that, please?
Yes, there are several reasons. So I think some years were just missed before the problem was taken seriously. So maybe there was the tendency to ignore that it may become a big problem, the migration issue of BPA and all the related substances. And then the second big problem is that it takes a very long time to develop this. As I've shown you in the presentation, there are many technical requirements which have to be fulfilled. So the material has to be long-lasting, so it has to withstand the storage and sterilization conditions for many, many years. Sterilization, of course, only for maybe 2 hours, but then it has to be in contact with the food and inert for many years. So this has to be tested, and one can think about testing conditions which shorten the testing period, but to make it really in a good way, one should go for several years of testing, or one has to reduce the storage time of the food cans. So that contributes to the long developing times. Yeah. But of course, once a problem is recognized, one should start to act on it, and that could have been done a little bit earlier.
Okay, so manage the issues before they become big problems. That's one of the messages, I think. Okay, great. So with that, I'd like to wrap up this webinar. Thank you everyone for joining. Thanks for the great presentations, Birgit and Maricel, and thanks to everyone for asking questions. And please, if you would like to share feedback with us on the content or the technical issues you may have encountered when trying to join the webinar, please let us know. As I said initially, we've recorded this webinar. We will make the recording available on our website, and please come back to our website to check for upcoming webinars that we will be hosting in the future. Thanks a lot and have a great day. Bye.
Thank you. Bye-bye.
Thank you.
Bye-bye.
Thank you.